Good afternoon, everyone. So we will start this week's tutorial session. I'm audible to everyone, right? Am I audible to everyone? Oh, okay, okay, sure. So my screen is visible, right? Okay. So this week's like we will be summarizing the week 7 lecture video content, and then look at some of the assignment questions. Okay. So the week 7 lecture videos were basically about how would you, like, after you have cloned your gene of interest, suppose something expressing a protein of your interest, and then you want to now take that protein out, purify that protein, right? So how would you start? Okay. So basic So first you have to definitely break the cells open. So for that, uh, that is cell disruption, which is the process where you obtain that intracellular or cytoplasmic, like whatever cellular contents, via opening the cell wall, and you take care that you shouldn't be disrupting any of its components. Okay. And this breaking the cells open depends on what is the type of the cell. There are fungal cells, bacterial cells, which have like cell wall as well along with the plasma membrane, and then there are mammalian cells which do not have any cell wall. So there will be now fungal cells have chitin around them, bacterial cells have peptidoglycan layer. So all the treatments, depending on what kind of cell is it, what kind of boundary it has, like cell disruption methods will vary. Okay. It should not be too harsh so that the product that we want to recover shouldn't be damaged. It should remain in its active form. And then cell disruption methods can be categorized into mechanical methods and non-mechanical methods. We will look at most of them in the subsequent slides. Mechanical methods are divided into solid shear methods and liquid shear methods, wherein you use some kind of solid medium or some kind of liquid uh, medium or surrounding the like medium, wherein you use the flow of liquid or the you use any solid material to disrupt your cells or break open your cells. And non-mechanical methods include your physical methods, chemical methods, and enzymatic methods. We will look into it one by one. Okay. So yeah, this is now cell disruption broadly can be divided into mechanical methods and non-mechanical methods. Under mechanical methods, it's solid shear or liquid shear. Solid shear has bead mill or press method, wherein you actually apply, you either break the cells open using glass beads, okay, like small spherical beads, or you press it. There's bench press method or French press method also, wherein you try to squeeze the cells through uh, like a certain kind of cavity, not exactly cavity, but through a passage wherein the passage is smaller than the diameter of the cell. Okay, like that. So it will eventually break the outer layer of the cells, break open the outer membrane of the cell. Okay, or cell wall in case of bacterial cells or plant cells. Okay. Then liquid shear can be categorized into high pressure homogenization or sonication, and there can be autoclaving, lyophilization, or microwaving, or thermolysis. Okay, like using heat to break open the cell. And under non-mechanical methods, we just saw it can be treatment using acids or alkali, like basically changing the pH of the medium, exposing the cells to changes in the pH. There can be osmotic shock, wherein you put in water to swell up the cell so that the cell finally bursts open. Then you can treat the cells with certain enzymes which interact with the composition components of the outer layer of the cell, so that uh, those particular structural framework gets degraded or dissolved. Okay. It's basically hydrolyzed, and then the cells can break open. So, like under mechanical ones, there can be mortar and pestle. Okay. I think in the lecture video it was shown, like there's like a kind of container, a bowl kind of thing, wherein you use, the spices that people use. You can use like that mortar and pestle is are there. These are like you put in that bowl kind of thing your cells, or uh, like mainly it's for breaking open cells which are tougher to um, lyse or break open. So what you basically do is simply grind the cells so that these are disrupted, and it needn't be in suspension. It is often done with plant samples frozen in liquid nitrogen. So what many times happen is, suppose we want to extract out proteins. So like in some laboratories, what happens, plant samples, suppose Arabidopsis, or any kind of wheat germ, like extract. Okay, so what people do is they just put it in a mixer grinder. Okay, and also, like other than mortar and pestle, this is another way of doing the thing. You put liquid nitrogen, okay, and just uh, using mixer grinder, you grind the whole plant tissue, okay, or plant leaves, whatever. You just put it in the mixer grinder and uh, grind the thing, and then so what will happen is, like when you pour liquid nitrogen, the whole cellular extract, or suppose the plant or leaves, whatever you want to study, from where you want to extract your protein, um, that particular tissue gets extremely brittle, so it becomes easier to really lyse it, okay, into powders or something like that. And then metabolites like suitable solvents can be added, suitable buffers can be added, so that though from that powdered extract, that powdered tissue extract, what you can take out your protein of interest, or any other macromolecule of interest that you want to purify. Okay. Then that this is what I was talking about. Either you use blenders, like after pouring liquid nitrogen, or mortar and pestle. Okay, blenders can be for a little higher amount, like higher quantity of the tissue. Suppose I'm talking here about plants, right? And in blenders you do high speed, you use high speed, which to disrupt the cell walls. It uses the same process as used by centrifugation, wherein you separate or concentrate the materials suspended in a liquid medium. Then you eventually get a pellet and a supernatant, right? And then there's bead beating. Glass or ceramic beads are used to crack open the cells. It is gentle enough to keep the organelles intact, but it will shear or break open the cells. And bead beating can be used with all kinds of cells. Just what actually usually people do is just add beads to an equal amount of cell suspension and vortex machine. If you remember, I think last to last, or in some previous tutorial class, I showed you the image. Like basically you can set the speed of vibration and disturb the whole suspension. Okay, and then that will mix it. The vortexing will eventually lead, like the glass beads as well as the cell suspension that you have will be mixing very vigorously. Like it will, the whole things, the content inside your tube will mix very vigorously. Eventually the glass beads will be beating against your cells, and then eventually will be breaking them open. Okay, so this is a mechanical form of lysis or cell disruption. So here what is happening, you can go to this, I've shared the paper also, the article. You can go through this article. From there only I've taken this image. So suppose there's a microalgae cell. you have put it inside along with your glass beads. These red ones are the cells and black ones are the beads here. Okay. Now, uh, you are vortexing the sample, right? And when you vortex it, there's a whirlpool, okay, inside of the solution, whatever is there, of the suspension that is inside the tube. Okay. So now, these beads, black colored beads, and the cells, the red colored ones, collide in like randomly. Okay. And then, so the different types of forces that the cells experience are like shearing force. Okay. This is like kind of moving past the cells and shearing it away, or it can be impact force, like directly hitting the beads, that directly hitting the cells, which can also break it open. This is the hitting force, which can also um, break the cells open and disrupt the cells. Okay, so these are like different. You can go through this article. This is kind of summarizing what happens, what are the possible forces acting on the cells when it gets collided with the beads. Okay. And then there's ultrasonication. This also we looked in like very briefly in the previous tutorial video. So here what happens, ultrasonic homogenizers work by inducing vibration. There's a titanium probe that is immersed in the cell solution. So suppose what happens is when you want to purify, so we looked at, like, you get a cells, you lyse your cells, maybe using a suitable, appropriate, optimized buffer. Okay. Now when you lyse your cells, the cell contents can come out. Okay. Now you centrifuge your cells, so you will get a cell pellet and a supernatant. Suppose I like my protein is soluble enough that it comes in the supernatant, right? Then that is one way. But before that, usually before this particular process, what people also do is, suppose people resuspend the cell pellet that you have pelleted down after overexpressing the like overexpressing your protein. Then what you like, after your secondary culture or subculturing, that cell pellet is resuspended in your lysis buffer, and then you go for ultrasonication, right? So that is the cell solution, or the cell, like resuspended cell pellet in your lysis buffer, that is being talked about here. Okay. So what happens is these probes produce like really high frequency sound waves, which leads to cavitation, wherein tiny bubbles are formed, and these explode like a bomb explosion, creating local shock wave and can disrupt the cell walls by pressure change. Okay. This is popular for disrupting plant and fungal cells. It is also like sonication in general we can use for disrupting bacterial cells also. Okay. Like routine protein purification, which is like the proteins which are expressed in E. coli cells, for getting for harvesting that protein out of the cell, you can people use sonication on a regular basis. Okay. But then you have to really optimize your conditions of sonication. So um usually what happens, I think there was a demonstration video also for like sonication and all. So that high frequency sound waves, that is given in pulses. Okay. Like, and the on time for that pulse should be like very very less as compared to the off time. So it should be given enough rest also. Okay. In between, like suppose people keep 3 seconds on time, 5 seconds on time, like for 5 seconds, or suppose 6 or 7 seconds, the sound waves will be generated, the frequency, high frequency sound waves will be generated. Then suppose 25 to 30 seconds off time will be there, when the sound, like that, those sound vibrations won't be there. Then so such cycles are given, so that on off cycles are given, like suppose 3 seconds on time and 15 seconds off time, like that. So over a span of maybe suppose 5 to 10 minutes of on time, and you have to set your amplitude also. Okay. So these are few of the parameters, and that has to be optimized also. Sometimes, your protein might not come in, or like you have overexpressed your protein and everything, you have optimized your expression, overexpression parameters also, but still when you are um, trying to express or purify your protein from large cell cultures, you might not see, or you might not get that much amount, your expected amount of protein. So out of so many reasons, one reason can also be that the sonication didn't happen well, that the cells weren't lysed properly, so that the cellular contents couldn't come out properly. Okay. So sonication parameters also have to be optimized or standardized. So see, this is the probe. Okay, this is the probe. This is your cell suspension here. And what happens is, suppose this is the electric probe, supplies sound energy, which gives rise to unstable growth of cavitation bubbles that we were talking about here, and causes size reduction. Suppose this is there. Okay, then ultrasound causes cavitation. Then this is again further broken into smaller droplets. Okay. So this is what happens. So this is the pressure of the sound. It is having like compression and rarefaction events. Okay. And due to this uneven and random compression rarefaction events, these bubbles are formed, which either grow eventually and then suddenly implode or explode. Okay. Thereby disrupting the cells. Then there's homogenization, which can be liquid based homogenization, wherein you disrupt the cell for small volumes and cultures, and cells are lysed by forcing the cell or tissue suspension through a narrow space that I was talking about. You can put in inside a passage, and shearing forces on the cell are similar to the bead method that we saw earlier. And homogenization can be performed by squeezing the cells through a tube that is slightly smaller than bead beating. Okay. Then there's freeze thaw cycle. Then there's freeze thaw cycle also. So here now, till here it was all mechanical ways of disrupting the cells. Okay. Now in freeze thaw, what happens is, like, here you are at like some external agent is being used here, but in freeze thaw, usually what happens is you mostly suppose in case of not only in case of proteins, just cell stocks, you can prepare cell stocks, right? Prepare cell stocks, and if you freeze the thing and then thaw the thing, so that cycle in between, when samples are freezing, then ice crystals begin to form, right? So due to the formation of these ice crystals, the formation of these ice crystals tend to damage the cells. So what happens is, in the competent cell preparation step, what happened, like, if you guys remember that competent cells are very fragile. So during when in laboratories we prepare competent cells, what usually is done is, when you have finally, like, after all the uh, divalent, like, after all the treatment with solutions containing divalent cations, what happens is, the these, like, after every treatment, all sorts of treatment, centrifugation and all, these cells are aliquoted. Okay, so immediately as soon as the cells are aliquoted, the suspension is aliquoted, we immediately put it in liquid nitrogen. Okay, which is at extremely low temperatures. So this process is called flash freezing. Okay, in a flash you just freeze the samples, right? So this, why do we do that? Because if we give this sub and then we store it in minus 80 degrees, okay, in refrigerator, which is the temperature is kept at minus 80 degrees. This is the usual protocol. So why do we do that? We could have easily just put the cells directly, right? Put the cells directly in the minus 80, right? So we don't want to give the sample or the surrounding medium, because there's water, definitely there's water content. So we don't want to give enough time for the ice crystals to form, so that it because the formation of ice crystals can eventually damage the cells, and competent cells being so fragile, we need to be, like, very careful while handling the cells. Okay, so the same thing is you applied here, like for freeze thaw. This is suitable for working with soft plant material and algae. Disruption or disruption is achieved using a series of freezing and thawing cycles. And uh, what happens is, freezing forms ice crystals. When we thaw that, it will expand upon thawing and cause the cell wall to rupture. Now, but then what happens is this freeze thaw cycle is a little time consuming. Okay, because you have to um, expose or let the cell uh, suspension go through a series of freezing and thawing cycles, and so it takes time to really disrupt the cells properly. Okay. Next comes microwaving or thermolysis. Thermolysis, as the name is suggesting, like using heat, you are trying to lyse the cells or break the cells open. So microwave along with autoclave and other high temperature methods basically disrupt the bonds within cell walls, and also this method should be used only when suppose you want to purify your protein and you know that your protein is unstable at higher temperatures, then this method shouldn't be used. Okay, so because it is definitely, if temperatures are higher, then it will eventually denature the proteins. Okay. However, uncontrolled amount of heat can easily denature or damage the target proteins and substances. Okay, so you have to again think about the properties of the protein you want to purify, and then apply this thermolytic method of disruption, cell disruption. Okay, then there's osmotic shock. What we do is like we keep the cell suspension in a, or suppose a cell is kept in a medium containing higher amount of solvent concentration. In case of osmotic shock, it's water, right? Hypotonic solution. Then what will happen is its solute concentration is low as compared to the intracellular environment, right? So what will happen is like water will try to get inside the cells, and so water moves into the cell, causing its volume to increase to the point that it bursts open, right? It swells up and bursts. And it can only work with animal cells and protozoa because these cells do not have cell walls. Okay. So, see, this is the osmotic shock that was being talked about. So, most of the mammalian cells have a plasma membrane with active transporter to maintain the osmotic balance. Now, maintaining an osmotic balance is an active process, which means it requires like certain like active transporters with and requiring the expenditure of energy, requiring the usage of ATP. Okay, prolonged exposure to the cell with hypotonic liquid wherein the solute concentration is low, and in fluids or liquids where the solute concentration is higher, that is called hypertonic with your reference solution, that is hypertonic. And if both the your solution of interest or your sample, so in this case suppose we are talking about the cell and the surrounding medium both have like relatively similar solute concentration, then it is isotonic media, like isotonic medium. Okay, so prolonged exposure to the cell with hypotonic liquid such as the water causes osmotic imbalance and ultimately causes cell lysis. So due to inflow of water, cells can swell and ultimately burst to release the products. Now, this is the Hof's equation wherein you can get osmotic pressure, which is pi here, and it has been definitely the osmotic pressure will be directly proportional to the concentration of solute and the temperature. Higher the concentration of solute, more water will gush in, right? Like move inside the cells, definitely. Okay, and temperature also. So, this is the equation pi is equal to RT C initial minus Ci, sorry, Ci minus C naught. Okay, wherein C naught is the initial concentration and I is, I think, the concentration at a given point of time. Okay, and also, so R is the yeah gas constant, T absolute temperature, Ci minus C naught is the difference between the total solute concentration between intracellular and extracellular environment. In this case, in this scenario. Now, each mammalian cell has differential susceptibility towards osmotic shock, depending on the properties. So, here, suppose they have given the example of red blood cells, which will be lysed with the addition of a tiny drop of water. But plant or bacterial cells are more resistant towards this osmotic shock or osmotic lysis due to thick cell wall, right? See, this is the control one, and this is the lysed cells where the contents have come out. the cell has been disrupted, and changing the color of the overall solution here. Okay. Now there can be electric discharges as well, where the cell is disrupted using electrical discharges in mammalian and other cells that are, surrounded by, or that have only have plasma membrane. Okay. So usually what happens is, this is an insulated chamber, a high voltage electric generator is there, shock waves are created, and suppose this is an example from this particular article. These are food samples, and then due to high voltage treatment, these are burst open, and then um, your you can get the products spilled out, like coming out of the cell of your interest, and then ultimately you can let that spilled out contents go via downstream processes to ultimately purify the product of your interest. Okay. Now, chemical lysis, like they are like organic solvents like alcohols, ether, or chloroform can be used to disrupt the cell wall. How? By permeabilizing the cell walls and membranes. Chloroform, we looked at uh, DNA isolation, RNA isolation, okay, like nucleic acid extraction, right? And this is handy to extract hydrophobic molecules like plant pigments, and EDTA, ethylene diamine tetraacetic acid, which is a chelating agent. Okay, it can be used specifically to disrupt the cell walls of gram-negative bacteria. Why? Because gram-negative bacteria have cell walls containing lipopolysaccharides, and these lipopolysaccharides are stabilized by divalent cations like magnesium and calcium ions. Okay, and EDTA being a chelating agent, it chelates these divalent cations. So what happens is, you use EDTA, destabilize the cell wall like composition. If like polysaccharides are stabilized by these cations, if these are taken away by adding EDTA, then the cell wall can be broken loose. Okay, but then you need to suppose for downstream processes, when you might be using certain enzymatic, suppose like as simple as enzymatic lysis, if you want to use or give any enzymatic treatment, many enzymes require these many kinds of metallic ions, right, for their functioning. So what happens is, you have to remove EDTA later, so that it doesn't really hamper your downstream experiments or downstream reactions. Okay, so usually it is done by dialyzing the EDTA away. Okay, wherein you slowly, uh, maybe via diffusion of buffer, what happens is, uh, the EDTA concentration is lowered. Okay, by using a dialyzing bag or dialysis bag membrane. Okay, there can be enzymatic lysis, which breaks open cells using enzymes that degrade cell walls and membranes. Depending on the organism, one can use cellulases for degrading cellulose, chitinase. In fungal walls, there is chitin, right? Bacteriolytic enzymes like lysozyme, which destroys peptidoglycan layer, mannanase or glycanase. Okay, and enzymatic lysis is useful if you want to isolate the protoplast, a cell without the wall. Okay, in such cases also enzymatic lysis can be very useful. Okay, now see, there can be alkaline treatment as well. What happens is, you use like really strong bases, or like provide a high pH environment, which is harsh but effective chemical treatment to lyse the cells. And alkaline treatment causes lipid saponification, which disturbs the lipid packing and affects the cell wall integrity, thereby making these walls loose. Okay, there can be detergent treatment as well, wherein you add a detergent solution to the cell, causing thereby solubilizing the lipid to form a micelle. Micelle are like amphipathic moieties, right? Wherein your polar heads are towards the outside. These ones, polar head towards the like outside, or like these are your normal cells, but micelles look like these circular moieties, wherein like spherical moieties, wherein your polar heads are outside and the hydrophobic tails are inside. Okay. The effect of detergent, what detergent can do is it can, like, linearly, it has been found there has been a linear correlation between the detergent concentration and the loosening of the cell wall. Okay, the detergent concentration which causes abrupt change in lipid solubility and forms micelle is known as the critical micelle concentration, or CMC. And there are many such detergents which are used, like SDS. This is also used in your electrophoresis for visualizing proteins, etc. CTAB, Tritonix, okay, digitonin, saponin, etc. You can also have controlled permeabilization, okay, controlled creation of pores on the cells. So this is like a partial cell wall disruption or permeabilization is achieved by using organic solvents like toluene. The organic solvent is absorbed by the cell wall, resulting in its swelling and ultimate rupture. But at low concentration, that organic solvent permeabilizes the cell wall without disturbing the cellular integrity. So this reaction allows to use cell as a reaction vessel to catalyze a reaction and get desired product. Many times, suppose when in cases when labs are working with very small nucleic acid molecules like tRNA. tRNAs are only 77 to 80 bases long, approximately. That is the average length. Now, if certain people like if for a particular experiment the required or the desired RNA is only tRNA, that small an RNA molecule, and the researcher doesn't want any other RNA, okay, to come out of the cell. So, but when these tRNAs are to be isolated from, like, suppose from bacterial cells, then chloroform is can I mean sorry phenol can also be used, which creates tiny pores on the cells, so that only molecules as small as tRNA can come out. Okay, like there will be further purification, downstream purification processes, definitely, but such small pores, like, it has to be optimized such that only tRNA, like, molecules in the range of, like, molecules of sizes ranging uh, like in the size range of a tRNA molecule, then only those molecules can pass through the, like, partially disrupted cells. Okay, pass, like, come out of the partially disrupted cells, and the other, like, bigger macromolecules can stay inside. Okay, so for tRNA purification also, a kind of partial cell wall disruption takes place wherein small tiny holes are created on the cells using organic solvent like phenol. Okay, see, like this. Hmm? Here, these are the tiny ones wherein the movement of metabolites can take place, or your macromolecule of interest can take place inside and outside of the cell. Okay, now so there can be ultrasonic extraction also, wherein ultrasound is used to create vigorous agitation, resulting in the extraction of your analyte, whatever sample you are interested in, from the matrix. Okay, there can be mechanical shaker also. This method depends upon the selection of solvent in which the solubility of analyte is higher. Okay, now the thing is, here we are talking about the extraction of like now your cellular contents have come out. Now how do you, out of all those of the whole extract, how do you really, you have lysed your cells, you have gotten the supernatant, suppose, after centrifuging. Now what will you be doing? Okay, because that particular supernatant is again having a range of uh, other biomolecules. Okay, so how do you separate your sample of interest out of the other biomolecules? So these are some of the things. There can be liquid extraction as well, wherein you partition the sample between two immiscible liquids. What happens is, your, suppose what you want to analyze, your sample of interest is more miscible in a particular solvent as compared to other solvent. Okay, and those two solvents are immiscible, but like what was happening in case of phenol chloroform extraction, right? So we were using the solubility of the DNA and proteins to separate it out, right? So kind of if we make that analogy, then here in liquid-liquid extraction, partitioning of sample between two immiscible liquids results in the extraction of interference-free analyte into the liquid in which its solubility is higher, like in which the liquid into which the sample of our interest is more soluble, sample will move towards that liquid, right? And then we can definitely see, you can always open this out and take the suppose there are two layers here and this is separating funnel, then you just open the thing, your sample of interest has moved into the upper layer, suppose. So you just open that knob and let the lower layer of the liquid pass out, and then just collect uh, the portion of the liquid which has our sample of interest, and then down like further apply other downstream processes to extract out your sample of interest. There can be solid phase micro extraction also, wherein analytes are present in the liquid sample, which get diffused and get absorbed on the fused silica stationary plate. Okay, like how it's done in TLC or thin layer chromatography, right? Coated on the fiber, these analytes are deadsorbed using minimum amount of solvents, or in general chromatographic techniques also. And then there's salting out extraction. What happens is you add salts, which results in the precipitation of analyte and thus extract out the analyte from the sample matrix. So what happens is it either salts or some other chaotropic agent. Okay, what happens is suppose your protein is there and your protein is interacting with the water in the surrounding medium in the solution in which it is present. Okay, now if you add salts, salts will be like will be more water loving as compared to the different other residues of the protein, thereby like affecting the interaction of the protein, like protein residues with the surrounding medium, thereby letting it precipitate out, and then you can extract your precipitate out of the solution. Okay, now this is from the lecture video only, how two molecules can be separated from each other. So three um organic molecules were taken: benzene, phenol, and aniline. Okay, benzene normal, like C6H6, and then phenol having one OH group and aniline having one NH2 group. Okay, these are similar to each other, but suppose there is a mixture containing these three compounds, then how would you really separate these out? So these are so what people do is they like try to play around with the chemical and physical properties, okay, of the compound which is present in the mixture. So the physical properties via which these can be separated is the molecular weight is different, boiling points are different. Okay, uh, definitely boiling points in cases where the compound in question is a liquid, or sample in question is a liquid. Freezing points are different, crystallization tendencies are different, solubilities are different, densities are different, right? And then chemical properties, the functional group, like phenol has OH, whereas aniline has NH2 group. Reactivity towards other reagent to form a complex. Okay. So for example, if you have a mixture of benzene and aniline and you would like to purify benzene rather than aniline, you can take the physical and different chemical properties of benzene into account and isolate it from mixture. So this was the example shown. Suppose these two are mixed here, then these are boiled. Okay, now these will have different boiling points. The one having a lower boiling point will evaporate faster, right? Now benzene has a lower boiling point than aniline. Then what will happen is benzene will form water vapor. Okay, it will go into now. What will happen here is there's a condenser attached. As soon as it goes, it will condense, and then benzene will be collected. Okay, and definitely what the whole mixture will have water content also. Now because of its lower boiling point, benzene will evaporate faster. It will be collected out in some other container. thereby separating it from aniline. Okay. Finally, you are having two separate solutions, right? Two or solvents. Now, here in this case, the distribution coefficient, which is denoted as Kd, is used to describe the distribution of a certain compound between two phases A and B. Okay. Now, concentration in phase A divided by concentration in phase B, like in liquid phase and vapor phase, suppose. Okay, in this particular example. Hmm? Now, suppose when you are doing thin layer chromatography, so how much of your sample of interest is in the mobile phase and how much of it is has bound to the stationary phase. So in that way you can get a distribution coefficient also. Okay. Now, what is chromatography? Chromatography is used to separate a complex mixture having different components into individual components, exploiting the partition effect, which can distribute molecules into different phases. Okay, owing to the different chemical and physical properties of the uh, mainly chemical properties of the like mixture in this case. Okay, distribution of a molecule between two phases A and B is given by the distribution coefficient Kd. And in most of the chromatography techniques, phase A is a stationary phase that I was talking about, or matrix, and phase B is the mobile phase or buffer, right? So the very first, like the very first chromatography technique was done by Michel Sweat, where he actually passed chlorophyll. Okay, chlorophyll along this matrix using ether. He used solvent ether, and then this whole chlorophyll pigment was resolved into different colors. Okay, I think the different components were like xanthophylls and something like that. Okay, and then slowly these were separated out. The color, like the pigments which constituted chlorophyll, were separated out. Okay, this was the very first chromatographic technique which was used. Okay. Now, in column chromatography, what happens? A stationary phase, the matrix here, this is the stationary phase, is filled into a cylindrical tube made up of glass or steel. And the mixture of analyte or your sample is loaded on the top. This is the sample mixture, and it runs from top to bottom. Now, how the distribution coefficient is exploited? Now, what happens is, suppose there are these red and yellow molecules are there, two different molecules. Now, what will happen is, eventually, because of their, suppose some solvent is going there. Now, in the previous case, suppose when this solvent was given, now this gray colored and red colored and green colored pigments get separated. But out of these pigments, suppose this gray one, which has moved the farthest, okay, then this gray one is more soluble in this particular solvent as compared to the red one, and the least soluble is the green one, right? So, or and also, like, the matrix will also play a role. Now, in this case, what will happen is, suppose for like in the mobile phase, if this is coming in the end, like this is coming out first, like towards the very bottom, this red one, so this is more soluble in the mobile phase, whereas these yellow ones are comparatively less soluble. Or it can be the other way around also. These yellow ones have uh, can interact with the matrix more as compared to the red ones. Okay. So what happens is, you inject this sample mixture, and as they will travel, these two will partition between stationary phase and mobile phase. And here in this example, as there is a huge difference in the distribution coefficient, y will be associated with the matrix and remain on the top of the column, the yellow one that is being talked about here, whereas X will move along with water or any other solvent. Here they have referred to as, like, they have used water. And at the end of the chromatography, X will definitely come out first because of its solubility, like higher solubility in the mobile in the mobile phase, or the interaction of the other compound that is at the higher level, then that is more with the matrix, that has a higher interaction with the matrix as compared to, or with the matrix or the stationary phase, as compared to this particular compound, or this particular and like um, yeah, this particular compound which has come out first. Okay, and the one that has like st- comparatively stable interactions with the matrix, or that like has more interactions with the matrix, that will elute out later. Now, what are some of the parameters that you should be considering when preparing a sample for chromatographic purification? So definitely when you are loading your sample, it should be clarified. It should be, you know, you can filter it, you can centrifuge it to really clear out for any possible contaminants, or any bigger particle that can actually clog the pores. Because see, in a chromatographic matrix, the stationary phase that we are talking about, those are packed, okay, using usually like agarose or any other matrix. Now or polysaccharide matrix, what happens is, if and there's a certain pore size or hole size, okay. Now what will happen is, if you don't clarify or lysate properly, then the issue might be it will clog your pores, okay. Or if you don't really clarify or lysate properly, then there might be interaction with the matrix, and those contaminants will bind to your matrix, whereas your sample of interest that you in like that you want, like your sample of interest to really interact with the matrix or the stationary phase, that interaction will be hampered because the interacting surfaces have already been occupied by your contaminating compound, okay. So you need to really clarify your sample before proceeding for chromatographic purification. Then you also have to check the stability of your target protein, right? Because you want to purify protein, and then when you break open the cells, there are numerous types of proteases that are present inside naturally, right? So you have to really inhibit those proteases, because it might hamper, definitely it might be your protein sample or protein of interest might be a good target for certain proteases. So you have to inhibit protease activity using, there's in the lysis buffer, people use PMSF, polymethyl sulfonate fluoride. There are certain protease inhibitor cocktail tablets that people can use in the lysis buffer. People can use benzamidine also, okay. And then you can play around with the pH, where to get the optimum range where your protein is stabilized, because like we were discussing the in the last tutorial also, your protein is made up of amino acids. Now amino acids have variable side chains, right? So how well are those side chains stabilized, it depends on whether the uh, H plus or OH minus concentration of the surrounding medium is higher or lower, right? What is the ionic state, right? Whether it is Zwitter ionic state, wherein it's neutrally charged, or it is positively charged or negatively charged. What are the reducing agents? There are stabilizing additives added. And then you should also be careful about the conditions for purification to work, right? Because mainly adsorption, we have to optimize the binding of target protein and minimize the binding of contaminants. These all are uh, you like mainly done using the buffers that we use, as well as few other physical parameters, like when there's the setup, like in a machine like FPLC setup, what happens is you also have to uh, you are able to control the flow rate, the uh, like what kind of uh, flow rate and pressure. So that can also affect the binding of your sample of interest or the protein of your interest onto the matrix. And then you have to be aware about, definitely whatever equipment is available, you have to optimize or tweak your experimental protocol according to that. And you have to take into consideration the practicalities and convenience, like what is the sample size, what is the available filtration or centrifugation equipment, etc. Okay. Now, centrifugation and filtration are standard laboratory techniques for sample clarification and used routinely when handling small samples. Okay, keeping the samples on ice is always recommended. Okay, even when purification is performed at room temperature. And when you centrifuge, we saw earlier also, it is able to remove most of the particulate matter like cell debris, and filtration can remove particulate matter. Membrane filters that give the least amount of non-specific binding of proteins are composed of cellulose acetate or polyvinyl fluoride. Okay. Either you do centrifugation, simply you separate out the pellet and the supernatant, or and suppose in case of or if a protein is prone to form aggregates, what also happens is, especially in case of gel filtration or size exclusion chromatography, what happens is if you centrifuge the thing, then your larger aggregates can settle down and you can just take the supernatant and inject the supernatant for flow through the size exclusion chromatography column, because the aggregates will definitely can harm or damage the matrix. Okay. For sample preparation before chromatography, filter pore size in relation to the bead size of the chromatographic medium should be selected. Okay. Now we have to consider the protein stability as well. In majority of the cases, we have to keep the biological activity intact after purification, definitely. Okay, retaining activity of the target molecule is beneficial when following the progress of the purification. And detection of the target molecule often relies on its biological activity, right? And um, in many cases, sample components can denature, which you can see by seeing certain precipitates in your tube containing the sample, or there can be enhanced non-specific adsorption on your chromatographic matrix. Okay, so ultimately all of these can impair the column function. Okay, like this precipitation can take place on the column matrix as well, thereby clogging the pores of the matrix and hampering, either clogging the pores of the matrix or interacting with the surface of the matrix, which is important for interacting with your analyte. Okay, so there are many advantages to checking the stability limits of the sample and working within these limits during purification. Now, proteins generally contain a high degree of tertiary structures, which are stabilized by van der Waals forces, hydrogen bonding. Hydrogen bonding is mainly between hydrogen atom and an electronegative atom like nitrogen, oxygen, fluorine, etc. Ionic interactions, hydrophobic interactions, and any condition capable of destabilizing these forces will denature or precipitate the protein. Okay. Now, peptides, proteins are much larger as compared to peptides, which are kind of fragments, right? Now, peptides have low degree of tertiary structure, and their native states are usually dominated by secondary structures, mainly hydrogen bonding, like we see in beta sheets, alpha helix. Okay, peptides can tolerate much wider range of conditions than proteins, and proteins are not easily renatured, but peptides can renature spontaneously. Usually, this is a general case scenario that we are discussing here. And proteins can form quaternary structure also, how we see in hemoglobin, like 4 subunits are there, right, forming a quaternary structure. Each tertiary subunit are coming together to form a like globular quaternary structure. So protein quaternary structures and protein complexes can give rise to additional challenges during purification, because complexes are often held by weak interactions and these require like mild or less harsh purification conditions. And then some proteins require coenzymes or cofactors to be active. So we have to add certain factors to make it active, or there can be some proteins which need to have a certain temperature to reach that particular like suppose not exactly proteins or something. Suppose in case of ribosomes, it is made up of ribosomal RNA as well as ribosomal proteins. So suppose for 30s ribosomes in vitro assays, it has been seen that it has to be activated so that it gets a particular confirmation, required confirmation for binding to the initiation factors. It has to be given a certain a treatment of an optimum temperature range, so that it gets it rearranges its helices to get an active conformation. Okay. So similarly, some proteins can require coenzymes or cofactors to be active. Like membrane proteins may need lipids from their natural environment in the cell membrane to maintain their native structure. Okay. Now, what is usually advisable to do is to perform stability tests before beginning to develop a purification protocol. Like how fast or how relaxed you can be while purifying your protein, it depends on how stable your protein is. Okay, there are proteins which you can, like, maybe many proteins, there are different steps of purification, right? One can use affinity chromatography, ion exchange chromatography, then go for gel filtration or size exclusion chromatography, or there can be just one step purification, that just affinity chromatography is taking place, or two step purification, affinity chromatography. So there are different combinations, there can be different combinations of chromatographic steps or purification steps to get your protein of interest. That again has to be optimized using different kinds of conditions, buffer conditions. Okay, but also it has to be kept in mind, if the protein is not stable, then suppose you have to finish, it is a two-step purification or three-step purification, you have to finish it as early as possible. But if your protein is quite soluble, like soluble as well as stable, uh, um, in pretty much on ice, then you can relax a bit and then perform your purification protocol, purification steps. Okay, then you have to monitor aggregate formation with size exclusion chromatography. How will you be able to monitor that? We will be looking at, I think, in the next tutorial session, wherein we will be reading about SEC. It provides a useful general stability assay for proteins. We should also be testing the stability to different pH ranges in steps of 1 pH unit between pH 2 and pH 9 of of our sample. Test salt stability with 0 to 2 molar NaCl or 0 to 2 molar ammonium sulfate in steps of 0.5 molar. Okay, including buffering agents as well. So buffering agents can be like trace, heaps buffer, there can be MES buffer, there are so many other buffers like that. Okay, which are responsible for maintaining the pH. Test the temperature stability in 10 degrees Celsius steps from 4 degree to 44 degrees Celsius. And at a minimum, the first test in the cold room and at ambient temperature 22 degrees Celsius. Like these are some of the suggestions or for someone to develop a purification protocol, which these should be kept in mind. Okay, definitely the protein stability should be checked. Test for protein stability and proteolytic activity. What people can do is, you can leave an aliquot of the sample at room temperature overnight and then run it on SDS-based gel to see if your protein has degraded or not. But before that, you should be knowing where your undegraded or your active form of the protein runs on SDS page. How does it, what is the gel profile of your active like functional protein? Then only you can really compare your degraded like stability of your protein, right? Now, to assay for proteolytic activities, it is advisable to run an SDS polyacrylamide gel to check the size of the target protein. Okay. Now, there are certain terms that uh, we will look at a longer list later in like after a few slides, but these are the more commonly used terms that you will come across. One is the chromatograph, which is the equipment that enables a sophisticated separation, like the whole setup. Then there's eluent, which refers to the fluid and or the mobile phase that is entering into the column, or the solvent that carries the analyte of our interest. Then there's eluate, which is the mobile phase leaving the column. Then the stationary phase, definitely the matrix or the immobilized phase we are talking about. This is immobilized on the support particles or on the inner wall of the column tubing, like silica layer in case of thin layer chromatography, right? So mobile phase moves in a definite direction, and the mobile phase moves through the chromatography column, the stationary phase, where the sample interacts with the stationary phase and is separated. Now, what is retention time? Like as the name suggests, this is the time taken by a particular analyte to pass through the system from the column inlet when you actually inject the sample inside the column to the detector under your set conditions or standardized conditions. Sample or analyte is the substance of your interest, sample of your interest, and solvent is the mobile phase, which usually in general it contains your sample of interest, but it can be the other way around as well. Now this is the chromatogram that you get. This is the visual output of your chromatographic technique. It shows the separation and how is the separation detected, how to read a chromatogram. There are different peaks or patterns on the chromatogram which correspond to different components of the separated mixture, right? So this is the ports or packing of the matrix, and then these two mixture, like this is the mixture of these two compounds which passes differentially through the pores and then separate out. So the very first one to pass through here is this triangle, like triangular shaped compound suppose, and this will elute out first, and the next one elutes out second. So detector signal is there, and the independent variable here is either time, and 2 being suppose larger, and or suppose it is interacting more with the matrix, then due to its lesser interaction, the triangle, the one number compound comes out earlier as compared to the second one. Either we can check, like plot the same thing in terms of time or the volume, right? Because your mobile phase is having your analyte sample, analyte mixture that is passing through. So how much volume? So higher the volume, that means later the thing is eluting out. Whatever sample or compound in question that is eluting out later, that means the volume will be larger. Volume of the mobile phase being used is larger. Now see, x-axis is the retention time, y-axis gives the signal here, and signal is proportional to the concentration of the specific analyte separated. So usually this is UV chromatogram. What happens is, how much is the concentration, the higher will be the peak. Okay, and lesser the concentration, lower will be the peak. Hmm? Now see, if this is your matrix, suppose your stationary phase and your mobile phase, this is the direction of the flow. Now how would you really represent the separation on a chromatographic column of when a mobile phase moves through it? Okay, now the solute that is there in the mobile phase, it slowly starts transferring from the stationary phase to the mobile phase at the back of the peak profile. Okay, um, and this is the profile of the solute concentration in the mobile phase. Okay, so this is there, and also when the solute transfers from the mobile phase to the stationary phase at the front of the peak profile, and this is the solute concentration in the stationary phase. So the separation, what exactly are the factors to rely on during separation of your analyte or your compounds inside the mobile phase or solvent? So there is intermolecular interaction between the two phases, in this case the stationary phase and mobile phase. Definitely the compound having more interaction, more affinity suppose towards the stationary phase will move slower as compared to the one that is not really interacting well with the matrix, or it interacts very well with the mobile phase. Okay, it can be the either, like it can be either way. Extent of dispersion of solute molecules over the stationary phase. Okay, then now suppose the interaction between suppose two compounds A and B are uh, of the same level of the same value, kind of same range to the interaction with the matrix. So then their separation will be like very poor, because they will be present at the same point. Or suppose something that is more what to say, has higher interactions with the mobile phase, then also it will, without interacting with the matrix, it will move along the mobile phase and the two compounds won't be separated properly. So we will have to take into consideration the different kinds of physical and chemical properties there. Okay. Now, two processes occur inside the column. The peaks are moved apart as a result of the relative affinities for the stationary phase that I was talking about, right? So if this has like lower affinity, so this is coming out first. This has like intermediate affinity with the matrix or the stationary phase, and this has the higher affinity. That is why it is eluting out at much later time or at higher volumes, right? Because it was interacting with the column. Okay, and this is the distribution system. So one is the peaks are moved apart as a result of their relative affinities for the mobile phase or the stationary phase, and the spread or dispersion of the peaks in question here in the chromatogram is constrained so that the solutes can be eluted discretely. If there is no separation that I was talking about just a few minutes back, like if two compounds A and B are not well, like they have similar affinities or interaction with the mobile phase or interaction with the stationary phase, then their separation will be trickier, and the discrete separation won't be there. Okay. Now, there's this thing called resolution in chromatography techniques. What happens is, in general, resolution is like how well two peaks are separated or two solutes can be separated by a matrix, chromatographic matrix or column. So resolution is a function of the selectivity of the resin. Resin is the matrix that has been packed into the column, and the efficiency of that resin to produce sharp, narrow peaks with minimal peak broadening. Okay, and resolution is expressed as this particular equation, wherein uh, this one VE1, this is the one that is being talked about, and VE2 is the volume here, which are the elution volumes for two adjacent peaks measured at the center of the peak. Okay, this is what I was talking about. Hmm? This is the volume axis, or it can be shown via time, but in this case, you can have the corresponding, because see, you when you pass your sample through the column, it has a certain flow rate which is denoted as x ml per minute, like milliliters per minute. So you can either have your x-axis on the, depending on like in terms of the time, minute or seconds, whatever, and or in terms of the volume, right? So yeah, at the center of the peak, and this W1 and W2 represent the width of the base of the peak. Okay, and definitely this, the difference between these two volumes represent the distance between the peaks, and this is the mean peak width of the two peaks. W1, half of W1 plus W2. Okay, this is like the definition of resolution that we are talking about. Okay, now what exactly is happening here? Suppose for a long column, or suppose for a like in case of paper chromatography or thin layer chromatography, there can be different layers or plates. Okay, so that the resolution is much better, so that the separation between two compounds is much better. So that suppose one compound has more affinity or more can interact much like has preferential interaction for the stationary matrix, then that is getting to interact more with the stationary matrix because we are placing more number of plates there. Okay, so that by the time that interaction is taking place, the other compound that doesn't have that much strong an interaction can flow out and elute out much earlier than the sample of your interest. Okay, or it may not be the sample of your interest. The one that is eluted out earlier might be your sample of interest, and the one that has stuck to that stationary matrix or is interacting more readily with the stationary matrix, that can also be your unwanted compound. Okay, so either way, there can be, you can exploit the chemical and physical properties of your mixture, so as to get your, like, singly purify your desired compound. Now, when less plates are there, then there's broader peak, there is less sensitivity and less resolution capability, because lesser plates means lesser interaction with the stationary matrix here. So if more number of plates are added, then there's narrow band, narrower peak, greater peak height, increased sensitivity, more resolution capability. Okay, so this requires columns with smaller particle sizes and instrumentation to minimize band spreading, right? So if band spreading occurs, then there's less interaction, like less resolution and less distinction between two different compounds, or it, like I'm talking about the simple case scenario wherein only two different compounds are there in your mixture. There can be n number of compounds in your mixture and you want only your sample of interest. So as to remove that, it's always good to have a, like, uh, increased resolution with a narrower peak. So these are, we won't be going through each and every one of them, just the very common ones used. In case of chromatographic techniques, there are various terms. The very first is adsorption. We have been talking about it, like, uh, like conceptually refers to binding and the process of interaction between the solute, for example, a protein, and the stationary matrix. Affinity chromatography is something which you employ to, like, using various types of specific affinity tags between target molecules, a protein and a specific ligand coupled to a chromatography resin. Last tutorial class we were looking at NINTA chromatography, right, using histidine and imidazole, right? That particular ring in histidine, which is, initially when you suppose a protein is tagged with histidine residue, histidine interacts with the NINTA column, and imidazole also has the same ring, and that ring, when it is used in, like, really high concentration, it can like interact more strongly with the NINTA matrix as compared to the tagged protein, like histidine residues on your tagged protein, and then your protein can eventually elute out. So that was affinity chromatography. Then this band broadening. This refers to widening of a zone of solute, for example, a protein, when passing through a column or a chromatographic system. What happens is it gives rise to dilution of the solute and reduces resolution. This is also known as zone broadening or peak broadening. Binding is definitely, like, either way, it's either adsorption or binding. Then there's binding buffer. Buffer, solution, or eluent used for equilibration of the column before sample loading. This is ideally used, the buffer condition that is used is usually the one in which your sample is suspended, okay, resuspended, or um, in which, like, the same conditions are provided to the column, so that when your sample is passing through it, it encounters the same conditions that it was already inside, right? It was already in, those conditions are provided again, so that, because pH, if in case pH differences are there or some other salt concentration is varied, then the interaction or the stability of the sample, suppose protein in this case, might be hampered. Then this binding capacity, the maximum amount of material that can be bound per volume of the chromatography resin. This also varies depending on what kind of resin is there, how good is the packing, okay, what is the pore size or bead size. Then there's chromatogram that we talked about, chromatography resin or the matrix. Column is the one that is packed. Column equilibration is the same that I was talking about using binding buffer. This is passage of the buffer or solution through the chromatography column to establish the conditions suitable for binding of the selected sample components, right? For example, to establish correct pH and ionic strength and ensure that proper counter ions or counter ligands are present. Suppose in case of nickel NTA, which I was talking about, the counter ligands are imidazole. right? Then there's pressure and column packing. The pressure is when, in case of FPLC or HPLC, what happens is there should be a certain pressure that the whole column can bear at its two ends, okay, because there is a certain flow. So if the flow or the pressure is not flow pressure is not maintained, it can damage the column, chromatographic column. Then you have column volume. What is the bed volume or the geometrical volume of the column? And this is very important to know because most of your buffers, uh, the whole purification protocol revolves around how many column volumes need to be there. You need to pass how many column volumes of a particular buffer you need to pass through the matrix so that appropriate purification can be standardized. Okay. Counter ion, ion of opposite charge that interacts with an ion exchange chromatography resin after the column equilibration, or counter ligand is the same thing in case it is ligand, the substances that interact with ligands of a chromatographic resin and can be displaced by a solute binding to the ligand. Now, dead volume, this is mostly, we will encounter this in size exclusion chromatography or in general because columns are packed and these won't be packed with 100% efficiency. There will be voids definitely. So this is the dead volume which is outside the packed chromatography bed and can be column dead volume or chromatography system dead volume. The dead volume contributes to band broadening. Degassing, this usually needs to be done in case of size exclusion chromatography wherein you remove dissolved air from buffers or solutions. If air gets inside your column matrix, that can be really harmful for the column as well as your sample as well. Okay. There is desorption, like opposite of adsorption, which basically refers to elution, release or removal of bound substances from the stationary phase or the matrix. Then there's efficiency measured as the number of theoretical plates. High efficiency means sharp peaks will be obtained. We looked at in the previous slide. Effluent is the mobile phase leaving the column or the eluate. Eluate is the effluent and eluent is the buffer or solution used during chromatography or the mobile phase. Elution buffer is used for elution or desorption of bound solutes. Elution volume is the volume of the buffer, elution buffer being used to elute the solute. And elution time, how much time did it require for removal of the solute that has been bound to the stationary phase. Flow rate that I was talking about, like volume per unit time. Volumetric flow, ml per minute, or linear flow rate, centimeter per like the height. So what is the measurement of this? Gives us a measurement of flow through a column. And then there's flow through gel filtration is size exclusion chromatography. Gradient elution and stepwise elution we will look in the subsequent slides, like within few slides only. Then there's HIC that we will also look at ligand ion exchange chromatography, ligand density related to ligand concentration, linear velocity, the matrix is the resin basically. Okay. Matrix is the non-functional base for the chromatography resin and it has a porous structure that provides a large surface that can be modified with ligands that introduce possibilities for protein binding. Peak broadening, what we saw in band broadening, it's the same thing. Peak fronting, broadening at the beginning of a peak, like a shift towards, like the peak is shifted towards the right side, then that is like peak fronting. Okay. Or and peak tailing is peak is shifted towards the left side, then there's a longer tail towards the right side. Okay, then that is peak tailing. Pore is the cavity in a chromatographic matrix, and pore volume is the total volume of the pores in a chromatography resin. Okay, resolution we talked about retention volume or time, and then there's reverse phase chromatography also, which is a method based on hydrophobic interactions between solutes. We'll be reading it in the subsequent slides. Then finally, there's sample loading, sample volume, how much of your analyte is going onto the matrix. Then there's selectivity also, which is measure of the relative retention of the two solutes in a column related to the distance between the two peaks. Okay. Selectivity, for a good selectivity, the distance between the two peaks should be optimum. It shouldn't be too less, so that two compounds are not really being resolved or separated out like very distinctly from each other. Solute is the dissolved substance, for example, the mobile phase, like your sample of interest basically. Stationary phase is your matrix. Void volume is the elution volume of solutes that do not enter the pores or interact with the chromatography resin, thereby passing between the particles in the packed matrix. Wash or wash buffer, basically use wash buffer for wash steps, wherein suppose your sample of interest is in the mobile phase. That mobile phase has passed through the matrix, and your sample of interest has interacted very nicely with the matrix, right? Now, there can be other non-specific interaction also, because there are many other things along with your sample of interest. Okay. Now, to remove those non-specific binding, a wash is given. Like, it again depends on how much is the column volume, how many steps of wash do you need to give. Okay. Washes do you have to uh, give. So those things depends on your column volume property of the solute as well as the matrix. So washes are given to remove non-specifically bound or other contaminants, and then elution actually takes place using a proper concentration of the ligand. And zone broadening is same as peak broadening, and yield is the amount of the target protein or any other solute or sample of your interest obtained after a purification step or after the entire purification, like multi-step purification. Okay. Now, where, like in real life, where can we really use chromatographic techniques? There can be pharmaceutical application wherein you are in drug development, etc. In food and beverage industries, in chemical industries, in forensic science, environmental analysis. Okay, this has been repeated twice. Now, depending on what is the property you are exploiting, there can be different types of chromatography. Okay, when the property under consideration, the characteristic under consideration is the charged state of your sample of interest or the protein, then ion exchange chromatography or chromatofocusing can be used. So ion exchange chromatography depends on the ionic state of the protein. Chromatofocusing also relies on the pi or the isoelectric point of the protein in consideration. Next, if you consider size, then this is size exclusion chromatography or gel filtration chromatography. If it is based on hydrophobicity, then it can either be hydrophobic interaction chromatography or reverse phase chromatography. Or if there is a specific ligand specificity or biorecognition, it can be affinity chromatography. All of this has been shown here, like gel filtration only on the basis of size. See the smaller ones that are there that actually get inside the pores, but the bigger ones can't enter the pores, so it will pass through the column at a much faster pace as compared to the smaller molecules. Hydrophobic interaction is like the same thing. There are certain molecules attached here, certain moieties attached to the matrix, and that interacts with the sample of our interest at a certain um, concentration of salt. And when we play around with the concentration of the salt, then this interaction can be either strengthened or weakened depending on what is the buffer condition being used. We will look at it. We will look at basically hydrophobic interaction and ion exchange in the subsequent slides, and probably the other ones in the next tutorial session. Now, ion exchange chromatography takes advantage of the unique relationship between net surface charge and pH of a specific protein. So, the interactions can be reversible between charged molecules, and oppositely charged IEX media are controlled to favor binding or elution of specific molecules and achieve separation. A protein having no net charge at a pH equivalent to its isoelectric point, that means a Zuiter ionic uh, state will not interact with a charged medium, and at a pH above its pi, a protein will bind to a positively charged medium, right? Because at pH above its pi, so at its pi value, it's neutrally charged, right? And at a pH above its pi, what that means is OH minus ions are more, so it will tend to take away H plus ions, right? Uh, tend to take away H plus ions. So what will happen is a protein will a protein will be negatively charged, right? So the protein will bind to a positively charged medium or anion exchanger. So there is some, so when we talk about anion exchange chromatography, then the matrix is positively charged. Okay, the matrix or the medium here or the stationary phase in consideration is positively charged, but it is called an anion exchanger because it is exchanging negatively charged moieties or sample. That's why this is an anion exchanger or anion exchange chromatography, but the medium is positively charged. Okay, because to this positively charged matrix, anionic compounds are getting interacting. Okay, but at a pH below its isoelectric point, what is happening? A protein will be having positive charge. So positively charged protein residues will be binding to the negatively charged matrix or cation exchanger. Okay. So there's sometimes confusion between these two, but when in case of anion exchange cation exchange chromatography, the matrix is negatively charged because it is exchanging the cations there. Okay, interacting with the cations. And also when you're passing your sample through a matrix during ion exchange chromatography, other types of binding may also occur, but these effects are really small and mainly due to van der Waals forces and nonpolar interactions. Okay. See, this is what has been shown here. So, I guess like most of you are aware about the titration curves. This is of amino acids. Okay. Now, so titration curves are basically showing the different, because due to the different kinds of functional groups attached to amino acids, these render them like there are different states wherein it can be positively charged, it can have multiple charged states, and amino acid can have multiple charged states as well at different pH uh, environment, at different concentrations of hydrogen ions. So now we see here that this is the pH range and this is the surface charge. So as the pH is increasing, what is happening is for a certain protein, for its pi value, like above its pi value or lower it like at a pH range lower it which is lower than its pi value, then it will be negatively charged, as it positively charged for a pH range lower than its pi value, and it will be negatively charged for a pH range above its pi value. Okay, what we read about earlier. So this is the same graphical representation of what will be the net surface charge as a function of the pH of the surrounding medium. Now, this is the general workflow of like it needn't be like any kind of chromatography. You should be equilibrating the column. Okay, then you load your sample or load your lysate. Then you wash it, give it a wash to remove any non-specifically bound contaminant or any other protein other than the protein of your interest, suppose. And then you finally, uh, there can be multiple elution steps or one step elution, whatever. You just elute it using salt, higher salt concentration or any other ligand. In case of affinity chromatography, basically elute out the things and finally like finally run past a very high concentration of salt or high concentration of a competitive ligand in your elution buffer to finally elute out whatever remnant amount is there in the matrix, absorbed onto the matrix that you remove out. Okay, but in case of ion exchange, what is happening here is it's the same thing, equilibration, which is equilibrated with the start buffer, or basically with the buffer that your sample is in. Okay, like resuspended in. So this is the matrix. In this case, the matrix has positively charged ionic groups attached. So basically here we are talking about an ion exchange chromatography. Okay, and this is an ion exchanger. The matrix is an anion exchanger because it is positively charged, right? Now this is a low ionic strength buffer. Okay, you have equilibrated the like your matrix, and then you apply your sample. Now what happens is when you equilibrate, this is the uh, now in the chromatogram, you like there are many kinds of values that you can get to see. One is the UV, which gives the one is the UV, which gives the amount of sample that is passing through, and also there's conductivity. So as the ionic strength is lower, it will be low, but as we increase the ionic strength, the conductivity will also increase. So that also can be seen in the chromatogram. Okay, so there are different settings to do that, but in general, you are able to definitely the conductivity will increase with higher ionic strength. And what happens is after equilibrating the column, you apply or load your sample. So what will happen in this case is your sample of interest is negatively charged. Okay, and there are other, along with your negatively charged protein of interest, there are other neutral or positively charged proteins also. So oppositely charged proteins bind to ionic groups of the matrix, becoming concentrated onto the column, and the uncharged proteins or the positively charged proteins won't really be interacting with the negative, like positively charged matrix with that much affinity or that much strength. So it will pass out. Okay, like it will be preferentially present in the mobile phase and elute out first. So uncharged proteins, or those with the same charge as the ionic groups, elute during or just after the sample application. Okay. Next, what I was talking about, there can be different steps of elution or one step elution, whatever, depending on what are your standardized conditions or standardized protocol. Slowly, you increase the ionic strength using a gradient which displaces the bound proteins here as ions in the buffer. The buffer is also strengthened eventually, wherein it also has like negatively charged moieties, which again interact more strongly with the positively charged resins, thereby displacing these negatively charged proteins that were bound to the matrix. So these negatively charged ions uh, compete for binding sites, which these ions are present in the elution buffer. Okay, and then further ionic strength is increased, amount of these ions in the buffer increases, competition for binding to the positively charged matrix increases, and hence the proteins are displaced. Eventually you see now most of the matrix is occupied at higher strength or at higher concentration of these negatively charged ions. Most of the matrix is occupied by those ions, whereas the proteins will now elute out. Okay, see, as the ionic strength increases, this blue line that is being shown here, the proteins start eluting out, right? So this is what is being shown here. And after elution, like at different steps, you basically wash it with final highest ionic strength wash, removes any ionically bound proteins before re-equilibration. Okay. Now, resolution, we looked at the resolution. This is what is distance between the peak maxima, right? We were looking at the two differences with the average base width of the two peaks. Okay, this is a typical chromatogram. This is volume versus UV. Uh, the higher the peak, the more is the concentration of the analyte or the sample. Okay, that has eluted out. Now, elution volumes and peak widths are measured with the same units to give a dimensionless resolution value. Okay, so resolution gives a measure of the relative separation between two peaks and can be used to further determine if optimization of the chromatographic procedure is necessary. Okay, now here suppose if the resolution is 1, then you have like 98% purity and 98% of peak recovery, provided the peaks are symmetrical and approximately equal in size as we see here. And baseline resolution requires that the resolution is greater than equal to 1.5, because at this value peak purity is 100%. Okay, and see, the if it is 100% peak purity, then that means the two peaks are very well resolved. There's no overlapping between two peaks as we see in the first figure here. Because of its 100% resolution, what is happening is the two peaks are very nicely separated. Okay, so suppose these two peaks are like there's overlapping between this part. So what happens is the volume that has eluted out, this in this part, the volume, the sample, whatever is eluted out in this part will have both A and B. Okay, but here it will be like extremely less portions of A and B. You will get pure A in the volumes prior to this area and pure B at volumes after this particular, collected after this particular uh, volume mark. Suppose this is, suppose 10 ml, suppose 10 ml or 50 ml, suppose. So before 50 ml you will have pure A, like maximum concentration of only A, whereas after 50 ml all these eluted volumes will have, or eluted fractions will have most of, like mostly pure B compound. Okay. So the more resolution is there, the more separation is there between two peaks, the more purity is there between two compounds. Okay. Like you, there are more chances of retrieving pure form of the sample of your interest. Okay. But again, one thing that should be kept in mind is a single well-resolved peak is not necessarily a pure substance but may represent a series of components which could not be separated under the chosen elution conditions. That can also be the case. Sometimes suppose there can be certain proteins which usually give a UV absorption of suppose 30. Okay, but sometimes it can be too sharp, only a single peak, extremely sharp peak that goes up to suppose 100. UV in this case is measured in MAU. Okay, suppose it goes up to 100, 150, something like that. So what can happen in that scenario is the whatever elution conditions we have given, the pH or any other thing, that might be favorable for other proteins also. Okay, and of similar size range. Okay, so then those couldn't be really separate, or uh, the other contaminating protein of similar size range can have similar interaction or similar affinity towards the matrix or the stationary phase. Okay, so then that might lead a, like like all the illusions, it will always try to stay with the protein or sample of your interest. Okay, so that will give us a well resolved single peak. But then again, because you have to check these fractions that have eluted out under these peaks on the SDS page gel to check really if you have got only your protein or sample of interest or there are other contaminating agents as well. Okay, now see what can be the different factors that affect the column efficiency. Particle size is a significant factor in resolution, and in general, the smallest particles will produce the narrowest peaks, okay, under the correct elution conditions and in a well-packed column. Okay, if there is uniform pore size distribution and small bead size, that will give narrow symmetrical peaks and minimal zone broadening. Okay, well-resolved peaks. Now this is what I was talking about, gradient illusion and stepwise illusion. Okay, gradient illusion is often used when starting with an unknown sample, as many components are possible, right? There can be, other than your protein of interest, many other components can be bound to the matrix or to the stationary phase. Now, if you give a gradient, like suppose we talked about NINTA purification, right? So you displace the histidine tagged proteins using varying concentration of imidazole. Now you don't really know for your protein of interest what should be the concentration of imidazole to be used so that the protein can be displaced completely from the matrix. Now what ideally you should be doing is, suppose usually in elution buffers, depending on what kind of protein is, we use a range of 300 millimolar to 500 millimolar imidazole. Okay. Now, uh, rather than using directly 300 millimolar to 500 millimolar imidazole, what people preferentially do in case the protein is you're starting with the new prep or new purification of this unknown sample, then you give a gradient, that is from 0 to 500 millimolar imidazole, you give a gradient. Then what will happen is eventually the gradient will be like, it won't be exact 500 or 300 millimolar. It will it will slowly increase from 0 to 500 millimolar imidazole. The concentration will slowly increase. Okay. So what happens is there are pumps inside. One uh, one pump will be in a very low imidazole concentration buffer, and the other will be in 500 millimolar imidazole buffer. Now the machine is given the command in such a way that suppose it will take 100% of low imidazole buffer first, and then slowly it will like 99% of low imidazole buffer, 1% of 500 millimolar imidazole buffer. So this way, slowly, the 500 millimolar imidazole buffer concentration will increase, and the low millimolar imidazole buffer concentration will decrease. And this is how there is a gradient, like a continuous gradient between 0 millimolar imidazole to 500 millimolar imidazole. Okay, this is an example just to show like how gradient elution works. Okay, so this will give us a high resolution separation or analysis, right? So this is what is happening here. And then after you get, now suppose you have optimized your unknown samples elution, now you know that okay, maximum amount of my protein is coming at suppose this range. So now you have this particular range wherein you know how much of your high imidazole buffer has gone in and how much of your low imidazole buffer has gone in. So you know exactly, you will be able to really calculate the exact concentration where your protein elutes. Exact concentration of the imidazole in the buffer at which your protein elutes out of the column. Okay. So then for the subsequent purifications, after this has been optimized, then you can always go for stepwise elution, wherein you get the exact concentration and elute out your protein of interest, because now you know that your protein is eluting at suppose you have used 500 millimolar imidazole, your protein is eluting at suppose has started eluting at 150 millimolar imidazole. Okay, so the next time onwards you don't have to worry about the whole gradient because that will use up a lot of your buffer. It will take up a lot of time as well. So the analysis will take a lot of time. But after you have known that, okay, this is the particular concentration of the elution uh, um, or the ionic concentration of the elution buffer, then this is what you just start with. You put that particular, suppose 150 millimolar imidazole, and start eluting out your sample or protein of interest. Okay. So this is the stepwise elution. When ion exchange separation has been optimized using gradient elution, changing to a step elution speeds up separation times and reduces the buffer consumption definitely, while retaining the required purity level. Okay. So this is what is happening. This is the stepwise elution. You directly give suppose 10 millimolar imidazole, then 100 millimolar imidazole. Okay, and finally, suppose exactly 500 millimolar imidazole, whatever was tightly bound to the matrix, to the stationary phase, that will elute out. Okay, and then you have to, there are definitely, you can't leave your column in that way because you have the column can be reused multiple times. So if you maintain, you have to wash the column properly, strip the column of uh, uh, like strip the column off from these tightly bound any other contaminating proteins or even your protein of interest, because the same column can be used for other proteins as well. Okay, and then you have to wash it with properly with distilled water, double distilled water, and store it in alcohol, right? Step elution can also be used for group separation in order to concentrate the proteins of interest and rapidly remove them from unwanted substances. The target protein is eluted in an enriched concentrated form. Okay. Or it can also be used to remove contaminants by choosing conditions that maximize the binding of the contaminants and allow only your target protein or your sample of interest to pass through the column and elute out first. So care must be taken to ensure that the binding capacity of the column is sufficient to bind all contaminants. Okay, so there are properties of the column that need to be taken into consideration. Okay. So then finally we have hydrophobic interaction chromatography, which exploits the ability of a strong interaction between hydrophobic group attached to the matrix and hydrophobic patches present on a sample such as protein, right? And that is what I was talking about, like salting out and salting in. You basically add salts to reduce the interaction of the protein with your uh, solvent or surrounding medium, and then you slowly increase the salt concentration, see here. And as you increase the salt concentration, um, after a certain point, the salting out, in the presence of more amount of salt, water molecules shielding the protein side chains are displaced completely with an exposure of hydrophobic patches on protein surface to induce precipitation of proteins or decrease protein solubility. And the phenomenon of salting out is modulated so that addition of salt induces exposure of hydrophobic patches on the protein, but then it shouldn't precipitate or aggregate the protein. Okay, this exposure facilitates the binding of protein to non-polar ligand attached to the matrix, and when the concentration of salt is decreased, the exposed hydrophobic patches on protein reduces the affinity towards the matrix and the protein is eluted out. This is the basic diagram. This is the matrix at high salt concentration. Water sol like it helps to helps the protein to really move away from, like, interact really less, relatively less, to have relatively less interaction with the mobile phase, and it interacts with the matrix. And when the low salt concentration is again lowered, then the protein again has more affinity towards the mobile phase, and it elutes out along with the mobile phase. Okay, now we will look quickly at the assignment questions here. Okay, so we will look at the assignment questions here. Now, which of the following is a physical method of cell disruption? Can anyone answer? Physical method we are talking about. Okay. Yeah, correct. It's sonication. Then we talked about osmotic shock method. Osmotic lysis can be employed in which case? Which of the following cells? No, because in case of bacterial cells, what happens is you have cell walls, right? So only water, like gushing in of water, movement of water is because of the thick layer of cell wall. What happens is water movement is restricted. But in case of mammalian cells, you only have a cell membrane. Okay, this is also a type of fungal cell. Okay, I think a species of yeast probably. Okay, so mammalian, this also has like additional layers, but mammalian cells only have plasma membrane. So osmotic shock method can be employed in case of mammalian cells, right? Okay. Now for sonication, uh, like high frequency sound waves are created. So a frequency of more than dash kilohertz is required for disrupting cells during sonication. Can anyone answer? 20 kilohertz. Okay, it is 20 kilohertz. Now, the same thing depending upon sonication. Sonication of cells suspended in liquid medium should be done in ice. Why? Can anyone answer? Just take a try also. It's okay. Yes, because these are like really high frequency sound waves, so it generates heat, and then your protein can be prone to denaturation at high temperatures, right? So to prevent that, the whole cell suspension is kept. Like we saw that the probe is inside the cellular, like inside the container, the beaker, right, having the cell suspension, but that beaker is again placed inside ice. Okay, and that's how sonication is done. Now, lipid saponification is a result of, like we just mentioned, uh, what will be the answer? So, you basically are destabilizing the lipid layer. So, saponification is occurs due to treatment with alkaline substances. Okay, so option will be C. See, suppose any lipid is hydrolyzed with a strong base, the process is called saponification. And the products of saponification are glycerol and fatty acid salts. Okay, like NaOH is used with saturated fats to produce hard soaps, and potassium hydroxide is used with unsaturated fats to produce softer, more liquid soaps. Okay, but the overall process is called saponification. Now, the enzymatic digestion of yeast cells would result in the formation of, can anyone take a guess or try? Yes, it's spiroplast. Option B. Now we just read about salting in and salting out. So addition of low amount of salt could facilitate the solubility of a protein. This effect is known as, yeah, correct. Now the isoelectric point of a protein is 6.5. So what buffers should we be using so that the protein could bind to a cation exchange column? If it is cation exchange column, that means the protein needs to be positively charged. Okay. So what will be the answer? Yes. So what will happen if the pH is 5.5? The answer is correct. Yes. So basically it will impart like positive charge due to higher amount of H plus ion in the medium, right? Comparatively higher amount of H plus ions, and positively charged protein will bind to negatively charged resin. Okay. Which of the following ion exchange matrix is an example of an anion exchanger? That means if it is anion exchanger, the matrix is positively charged. Okay. So what will be the answer? Yes. It is diethyl aminoethyl or DEAE. Okay. Now, just a very small information, like DEAE can be, again, it can be DEAE saccharose or DEAE cellulose. So one can, resolve proteins over a high pH range, like broader range of pH, and one can um, resolve proteins in a narrow pH range. Okay, so like these again, so even if one is anion exchanger, it can have variations depending on its charged form in case what is the pH of the surrounding medium. Okay. Now, a mixture of four proteins with their charges in buffer of 7.4 are as follows. Okay. And in case of anion exchange column, that means the um, if the column is anion exchange column, then the matrix is positively charged, right? So the protein that will bind strongly to the matrix is, yes, okay, having the negatively charged residues will bind more strongly. Now, which of the following property of a protein is exploited in gel filtration chromatography? Yes. So what is the other name for gel filtration chromatography? Yes, yes. So it is also known as size exclusion chromatography because you separate the components based on their sizes or mainly surface area. Okay. So yes, option C. Now a chromatographic column has a length of 100 centimeters having a height of 1 centimeter of the distribution plane, and the number of distribution planes of the column is, it is given by length by height. Okay. So it will be option C. So this is from the lecture video only. See, in a column chromatography, the resolution is directly proportional to the number of distribution events, like number of plates there are. Okay. And each thin plane of column matrix participates in distribution of molecule. Now if the height of the plane is h and length is l, then the number of distribution plane in a column is given by this. Okay. So this is straightforward, like L by H. So 100 will be the answer. Okay. So number of distribution plane in a column controls two parameters. So if the distribution plane, number of plates are going up, it will allow the analyte definitely to interact more with the matrix and it will travel for a longer period of time. Thereby it will increase the distance between two peaks. Okay. And if the number of distribution plane is higher, or it goes up eventually, it will reduce the width of the base of the peak, right? Because what will happen is, if it is resolved very nicely, two compounds are resolved very nicely, then if it is resolved nicely, then one compound will just elute out over a small volume, right? And the peak will be narrower. Like two compounds will be separated very nicely, resolution will be good. Okay. So as the number is increasing, the peak width is decreasing. See, as the number of distribution plane is increasing, the width of the peak is decreasing here. So the number of distribution is an indirect way to measure the column efficiency, and definitely higher number is desirable for better separation. Okay. Now, the distribution coefficient or KD value of XYZ are 1, 5 and 10 respectively, and these have been mixed and passed through a column. So what will be the order of their elution? Can anyone answer? So if the distribution coefficient is 1, 5 and 10, which means lower distribution coefficient means it is interacting lesser with the stationary matrix and it is interacting more with the mobile phase. Okay. So if it is interacting more with the mobile phase, it will come out fast. Okay. As compared to the other ones having higher values of distribution coefficient. So if x has 1, y has 5 and z has 10, that means z is more like distributed over a, like very nicely over the distribution plates on the matrix and interacting more readily with the matrix as compared to the other components. So it will stay on the matrix for longer time and elute out at the end. Okay. So the order of the elution will be, x will come first having lower one. Okay. Like lower KD, like lowest KD among the three, and Z will come in the end having the highest KD among the three. Okay. So the option will be XYZ. Now the cell disruption in a in a bead mill and release of products follows what kind of order kinetics? Does anyone know this? Yes, it is first order kinetics. Okay. So the rate and degree of how much the cell is opening up, how much are we disrupting the cell, it depends on the cell type, thickness of cell wall, localization of the product, how much speed are you applying, okay, what is the bead size, how much of like what is the amount of beads you have loaded, all these things, what is the temperature, how long are you uh, like incubating the beads as well as the cell suspension. All of these things affect the cell disruption. And this is the first order kinetic equation. wherein the C max is the concentration of product that can be released from a given amount of cell suspension. C is the concentration of product released at a given time t. K is the first order rate constant. Okay, and value of K depends on the type of impeller, what is the size of the bead, and how much of your cell lysate or sample has been loaded, what was the speed of agitation. Okay, like if the speed is higher, then the number of times the beads as well as the cells will collide with each other, that will be greater. Okay. Oh, yeah. This is the last question. Like this is like it is directly asking the equation for dynamic pressure, which is experienced by cells in a high pressure homogenizer. So what happens here is, in general, in case of high pressure homogenizer, we are talking about the liquid flow. So there's a theorem called Bernoulli theorem, which talks about the pressure of the, which takes into consideration the pressure of the liquid at two ends and the kinetic energy or movement. So the energy density, so kinetic energy is half mv square. So energy density or the dynamic pressure is ps half, if we are talking about the density here, then half mv square divided by the volume. So m by v will give density of the liquid. Okay, density of the liquid or the fluid in this case. So it will be rho. Okay, so it will change to p is equal to half rho v square. Okay, this is like half into in the numerator only rho v to the power 2. Okay, so ps is the dynamic pressure, v is the velocity, and rho is the density of the fluid. Okay, so option will be option A, half rho v square. So this is it for today's tutorial session. We will meet again the next Sunday. Okay then. Thank you for joining. See you all in the next tutorial session. Thank you.